I'm going to start off with a little bit of an outline of my agenda. What is plant health? I'll speak to this in the context of local Canadian terms. How is plant health measured in our environment with the crops that we're focused on? We'll talk about actual Canadian plant health research-based field results. So first off, what is plant health? Well, if we put this in a commercial context, really to be of value, any plant health benefits that we're discussing or focusing on should relate to parameters that we can measure. By that I mean measurements that we can describe with specific units. They should not be subjective measurements. We all like to talk about things like we have a lush crop or a vigorous crop, but in the end, what does that really mean? These parameters that we measure should in the end result in increases in grain yield or quality, and both Kent and Wolfgang referred to those two different parameters. So the obvious question, how does plant health influence grain quality and grain yield? Well, if we think of quality first, clearly if you've been in the fungicide discussion, we think of classical fungicidal crop protection. I'm not going to focus a lot on that today. That's a given in this discussion. We all know that, for example, we use fungicides for control of plant diseases. We can talk about genetic disease resistance. If we're a plant breeder, of course, breeders introduce a genetically based disease resistance into plants. But we can also have systemic acquired resistance that we can influence, as Wolfgang showed in a number of different venues. And so that is something that is definitely plant health related, and we'll talk about that a little bit. And then secondly, in terms of quality, we can also look at influences on protein, specifically as it relates to cereals, or protein and oil content in the case of some of our oilseed crops that we deal with. The major parameter, of course, you as agronomists dealing with growers, you often relate to this directly. The bottom line often is yield. And how can we influence yield with plant health? Well, first off, we think of partitioning of energy or carbohydrates. If you can move, assimilate, or sugars within the plant as the plant matures from the vegetative matter up to the head, clearly you can influence yield in that respect. You can also increase your yield potential by influencing photosynthetic area, essentially the solar panel. Or you can increase photosynthetic efficiency. So Wolfgang referred to the quantum efficiency of the photosynthetic process. And particularly if you can increase one or the other or both of those over time, you have the ability to build greater yield potential. So I want to focus a little bit on how we measure those types of improvements related to plant health. I'm going to get to field results, but before I do that, I'm going to start out with some lab results. As Wolfgang clearly illustrated, it's much easier to show benefits from plant health related products or compounds when you're under controlled conditions. So I'm going to relate to a study that was conducted in Monheim by Anne-Sophie Heinze, and Wolfgang referred to some of Anne's work. This is another study that she has done. It is in a greenhouse. It is under controlled conditions. This allows us to control disease and insects, and so we can relate our results then directly to plant health benefits. So we're looking here at fungicides, in this case demethylation inhibitors or DMI fungicides, group 3 by the FRAC classification. We've been talking about these extensively already in this session. So we have Procero. This is the same formulation that we have in Canada, the EC250. 2 reference DMI fungicides, and then tebuconazole or Folacure EW250. The evaluations that were conducted are photosynthetic activity measured in terms of quantum efficiency, chlorophyll content, and we'll talk about thermography and yield. So Wolfgang mentioned how photosynthetic efficiency and specific quantum efficiency can be measured using chlorophyll fluorescence and fluorometers. Essentially, solar energy, we want to go as much as possible into photosynthetic activity. Most of it is, but there are non-productive processes relating to heat and fluorescence. And by measuring those, we can determine quantum efficiency, which, as described here, is the efficiency of photosystem 2 in capturing incident light and converting that into photosynthate. So what we have on this slide is now at soft stow stage in wheat. On the left-hand side, quantum efficiency, or the efficiency of photosystem 2, it's now measured as just an index value for the flag leaf and the penultimate leaf, which we know contribute in a significant way to the filling of the head. And we can see that clearly Procero has a very high level of photosynthetic efficiency. We can also compare that to chlorophyll content, which can be extracted quite readily out of leaves and evaluated in a lab scenario. And in the same two leaves, flag leaf and the penultimate leaf, we see that there is a high level of chlorophyll content agreeing very closely with the quantum efficiency. The second evaluation that was mentioned on the study introductory slide referred to thermography. Essentially, think of this as a photo that represents the amount of heat being radiated by the plant. So the very, very light colors, or white, represents high temperature. So that's warm tissue. The blue tissue represents cool leaves. In this case, you can see this quite evident in the Procero treatments. And the four different boxes here are essentially just four replicates with the same treatments. And you can see that plants treated with Procero are cooler. That means that there is a transpiration taking place. There's water still being uptaken. There is photosynthesis still happening in those leaves. They are still photosynthetically active, and therefore they are fixing, assimilate, and building yield potential. And they are therefore less senescent than the plants that are not treated with prothioconazole. So finally, those previous parameters ultimately, to have greater meaning, need to result in increased yield potential, and that's what these charts show, both in terms of number of grains per year as well as kernel weight. And as a result, you can see the higher yield provided by Procero, and Folicure also looking quite promising in that regard. So clearly the photosynthetic efficiency and the increased chlorophyll content confirmed by the thermography results in higher seed yield. There's a strong agreement between those results. So that's in the greenhouse under controlled conditions. Wolfgang mentioned some of the tools that we can use under field conditions to measure plant health as well, with reference to normalized differential vegetation index, or NDVI. So Wolfgang's presentation described how NDVI is calculated. I won't go into any further detail here, but I'd just like to point out for reference what that index and the numbers in that index refer to. So if you have a value near 0, it essentially means there is no green vegetation. The highest value possible from a plant stand is in the vicinity of 0.9. And I'll show you what that looks like in terms of a crop in a field. So these are actual photos of research trials from Western Canada. And you can see with the various slides the different NDVI values, starting from a newly emerged crop at a very low NDVI. Maximum NDVI is usually reached at early head emergence. Flag leaf is now out. You have maximum canopy development. And from there, you start to get a reduction in NDVI as you have senescence. Initially, it's just some of the lower or mid leaves in the canopy. Eventually, at full maturity, you have very low NDVI again, but that correlates very closely with the green matter, or lack thereof in this case, at or near full maturity. So to be of real value in terms of a predictive measurement, NDVI needs to be correlated to something of commercial importance. And there's been a number of different studies published that show strong correlation to a number of different parameters. The first one here, which is shown at the chart on the right, is percent green area, very strong correlation, R value of 0.91. But there's a number of other parameters here that are of interest. We talked about chlorophyll content, but ultimately what we're driving at is grain yield down in the bottom, and there have been correlations drawn in published literature. And in fact, as a result of work that's been done, we know that NDVI is already being adopted and utilized on a broad scale basis by public institutions. This is showing Statistics Canada, which now for approximately 3 years has used NDVIs in their crop condition assessment program to help in providing predictive values for crop yield potential. And I'm sure a number of you have followed the Western Producer as one of the public agricultural newspapers. And NDVI models go into predicting crop yield, which now are in public press. If we bring that closer to home, though, in terms of research work in Western Canada, it's almost 10 years now since some of the first pioneering work was done by the late Dr. Guy Lafond from Agriculture and Agri-Food Canada at the Indian Head Research Station. And he was one of the first ones in Canada to draw a relationship between NDVI and grain yield in spring wheat. And most of his work was dealing with fertility, but showed a clear correlation between NDVI and yield. Since then, we've been using green seekers for a number of years in our research program at Bayer. Wolfgang mentioned a PhD student that he supported and helped sponsor. We were fortunate enough to provide a home for that PhD student to conduct his research program. And this was from 2012. And so this is from Mark Rist's program here in southern Alberta. And he was clearly able to show a correlation between NDVI assessments taken with the green seeker, shown at the right-hand side, and yield. And just as a sidebar, I actually do have a green seeker unit. If there's time and interest following the presentation, I'd be more than happy to show participants how that works, just in terms of functionality. But the take-home message from this slide is that NDVI can be a useful predictor of yield potential in small plot research. So now what I'd like to do is give some examples of commercial product and how that relates to plant health yield enhancement in spring wheat. I'm going to show research results from Western Canada over a period of just very recent time. So if you've been in the fungicide session, Troy showed you a slide relating to a large number of field scale research trials conducted over the last 4 years in Western Canada. So we have 89 trials between 2009 and 2013. And the slide showed medium to high leaf disease pressure on the right-hand side. And as expected, no surprise, there's disease present. We're applying fungicides in this case, and we see a yield increase. The numbers here are shown in bushels per acre. But what's interesting is when you look now at the 0 to low leaf disease pressure, and you see that there's still a very significant yield increase. And you wonder, yes, you can never be at the total exclusion of pathogens in a field environment under practical conditions. But in many of these trials, there was no observable disease pressure. And the logical question is there something more than just disease control that's taking place? Clearly, to answer that question, you need more controlled conditions. So we've set out to do the best job possible in dedicated plant health trials to evaluate the effect of Procero on yield. And we've set out to make these trials disease-free. And you might say, how can you ensure that under field conditions? So we use best agronomic practices. We're all agronomists here. I think we all understand what those would be, but solid crop rotation, a high quality seed, disease resistant cultivars. We'll even go so far as to use a blanket spray of a fungicide of a different mode of action if there's any disease incidence to try to maintain that disease free as much as possible. And the result over these 10 trials in 2 years is that we see a 3.3% yield increase from Procero. And over on the right-hand side, the right-hand axis, you see AU-NDVI. That refers to area under the normalized differential vegetation index curve. One of my first slides, I referred to the value of increasing your solar panel or your leaf area over time. What we've done is taken multiple NDVI assessments. And essentially what we're looking at is the total volume or area underneath that NDVI curve over time. And in fact, we can see an agreement between the NDVI over time and yield. So nice agreement there. A logical question as agronomists dealing with grain, we know that often if you see a yield increase, there is an offsetting decrease in grain protein, particularly in wheat. And so what we did is we conducted protein analysis on these samples from these same trials, and we see that there was no effect on grain protein, which is very positive. It's essentially win-win. You can also notice from the numbers here that these were very high level of fertility. These trials were conducted under really top level management in terms of fertility and all agronomic practices as a whole. So that was fungicides. I'd like to spend a little bit of time now talking about stress shield. So Wolfgang spent a fair bit of effort describing how imidacloprid works on a biochemical basis on plant metabolic pathways. And what I'll show you is some field results related to imidacloprid. So right off the top, first and foremost, we are dealing with, in this case, a seed treatment insecticide. The reason we use it on a commercial setting is for things like control of wireworm. So we're looking at some research results here from moderate to heavy infestations of wireworm. We're picking up 5 to 10 wireworms per bait ball when we're doing our monitoring. And you can see, as expected, goes with commercial experience, that when you use a seed treatment insecticide, there's a pretty substantial increase in plant stand. In this case, roughly a 20% increase in plant stand. We're not killing wireworms, as we know. These are CNI insecticides, but we are protecting the crop and maintaining our plant stand, and therefore expecting to maintain our full yield potential. But what's interesting is when you look at a large number of trials comparing these same treatments and look at the yield, we see some differences showing up. So even though the plant stand from the various seed treatment insecticide were similar, we're seeing a yield difference. So the obvious question is, why? So we set out to evaluate these same treatments, looking at situations where there's no insect feeding pressure. And you note the comments in red up at the top, no feeding pressure. So how do you make sure there's no insects? Well, that's in practical terms very difficult under field conditions, but what we do is we select fields where there's no known history of insects. Where we set out bait balls, we can pick up no activity on wireworms, for example, migrating into a trap. And so we know that there's very low or no insect feeding pressure in these trials. And what we see under these circumstances, in fact, is that when we apply stress shield or imidacloprid, that we see increased plant stand relative to the fungicide check. When you take that next step farther and you look at NDVI from these same trials, we see that as you would anticipate, there's increased vegetation index, increased green leaf matter. That would be expected if you have an increased plant stand, and that's in fact what we observe. And then when we take that to yield, you can see that it does translate into yield. So we are seeing a nice little yield increase from the base fungicide. There are background pathogens in the soil, and so we're seeing that yield increase. But when we add the imidacloprid or the stress shield, we're seeing in this case a nice little about 4.5% yield increase on top of the base fungicide. So over a larger number of trials, what we've done since 2011 is we've evaluated stress shield in trials set out to avoid both insect and pathogens. So the idea here, this is plant health style trials, and we're gathering as many data points as possible to get as reliable a data set as possible. And what we're picking up is about a 3 or 4% yield increase when we have situations that we're controlling both disease and avoiding soil insects. So this is attributable to the stress shield benefit from imidacloprid. So that's commercial products. That's products that are commercially in the market that everybody's familiar with, and that's our field research experience from Canada.